Welcome back friends. Welcome to another video from Sci2K. In this video tutorial, I'm going to discuss division of cell. How does cell divide into two daughter cells? Why is cell division important for living system? So let's understand it in detail. Stay tuned and continue to watch this video. Cell division. It is a biochemical process in which a parent cells divide into two daughter cells. Walther Flemming was first to document the details of cellular division. Cell division is important for different reasons in living organism. In unicellular organism like bacteria, the production process occurs by binary fission, while in eukaryotes, cell division is important to maintain chromosomal number, maintenance of Nucleo-cytoplasmic Ratio, repair, healing and regeneration. It also helps in formation of gametes in multicellular organism by meiosis process. But before going to divide, every cell need to prepare its cellular content for division process. This preparatory process is very important in cell division that I'll discuss in later part of video. First, we will see what is cell cycle. Cell cycle is a cycle of stages that cells pass through to allow them to divide and produce new cells. The two main parts of cell cycle are mitosis and interphase. Mitosis is a phase of cell division during which a parent cell divides to create two daughter cells. The longest part of cell cycle is interphase, the phase of growth and DNA replication between mitotic cell division. Both mitosis and interphase are divided into smaller subphases which need to be executed for cell division, growth and development. Interphase consists of three distinct stages during which cell grows, produces new organelles, replicates its DNA and finally divide. One is G1 phase, second is S phase, third is G2 phase, and mitosis consists M phase. There is another phase that is known as G0 phase or resting state of cell. In G1 phase, the cell produces many essential materials such as proteins and ribosomes. Cells that rely on specialized organelles such as chloroplast, mitochondria make a lot more of those organelles during G1 phase. The cell size may increase as it assimilates more material from environment. This allows the cell to increase its energy production and overall metabolism, preparing it for S phase. S phase is a synthesis phase in which DNA replicate, and we can say that 46 chromosomes replication. When S phase is completed, the cell will have to complete sets of its genetic material. This is crucial for cell division as it ensures that both daughter cells can receive a copy of the blueprint they need to survive and reproduce. Just like the first G1 phase, the G2 phase is characterized by lot of protein production. During this phase, many cells also check to make sure that both copy of DNA are correct and intact. In M phase or during mitosis, the parent cells goes through a complex series of steps to ensure that each daughter cell will get the materials and it need to survive. Once the material are properly sorted, the parent cells divide down the middle, pinching its membrane in two, and the mitosis followed by cytokinesis or cell separation. After being born through mitosis, there are some cells which are not meant to divide themselves to produce daughter cells, like neurons that do not divide. These cells are in resting state. So instead of going into G1 phase after being born, neuron enter a phase that is known as G0 phase. This is a metabolic state meant only to maintain the daughter cell, not prepare for cell division. So what is the importance of cell cycle? The importance of cell cycle can be understood by doing simple math about cell division. If cell did not grow in between division, each generation of daughter cell would be only half the size of the parent generation. This would become unsustainable pretty quickly. In order to accomplish this growth and prepare for cell division, cells divide its metabolic activities into distinct phases. So first we will see the process of mitosis. This type of cell division gives genetically identical cells. in which the number of chromosomes is maintained. That's why it is known as equational division. It occurs in all multicellular organisms or in somatic cells. In mitosis, one round of replication followed by one round of segregation. Let's understand the distribution of chromosomes and amount of DNA during cell division. After mitotic cell division, there is no change of amount of DNA and chromatin number with respect to daughter cells and parental cells. So let's understand the distribution of chromosome and amount of DNA during cell division. Here, X represent the number of chromatin present in cell, while C represent the amount of DNA present in cell. As you know that somatic cell contain 23 pair of chromosomes. For better understanding, here I am showing only 2 pair of chromosome. Chromosome number 1 and chromosome number 2. In G0 phase, total number of chromatin is 4 and amount of DNA is also 4. So these cells are diploid cells. I have written 2X, 2N, 2C. Once cell goes into G1 phase to S phase, what happens? In S phase, it replicates genome. At that point, total number of chromatin is still 4, but amount of DNA is 8 because of doubling of whole genome. Then cell goes in M phase, where spindle fiber pulls the chromosome at periphery. Here, amount of DNA are still 8, while chromatin become 4x because sister chromatids are separated. After cytokinesis, the daughter cells contain 2 pair of chromosomes, same as parental cell. That's why this mitosis is known as equational division. Amount of DNA and chromatin are same in both parental and daughter cells. But the distribution of chromosomes are different in meiosis process. Let's understand it. Meiosis is a process where a single cell divides twice to produce 4 different cells containing half the original amount of genetic information, and that's why it is known as reductional division. In this process, one round of replication followed by 2 rounds of segregation. In first round of segregation, separation of homologous chromosomes, while in second round of segregation, separation of sister chromatid. This type of cell division occurs in reproductive cell, sperm in males and eggs in females. So in G0 phase, the 2 pair of chromosome are present, total number of chromatin is 4 and amount of DNA is also 4. One cell goes in first round of meiosis. The homologous pair of chromosome are separated into 2 daughter cells. If you can see, one chromosome present here, another pair of chromosome are present here. Each cell contains 2 chromosomes as a haploid, while total amount of DNA is 4. After second round of meiosis, 2 daughter cells are divided into 4 haploid cells which contain half of chromosomes and half amount of DNA, that is 2. This reductional division is important because it ensures that all organisms produced via sexual reproduction contain the correct number of chromosomes. It also produces genetic variation by way of process of recombination that we will see in later video. If you like this video, please like, comment and subscribe my channel. We will see you in next video. Thank you and keep learning.